Hello, so this is my poster presentation titled Integrating Current Analyses of the Breast Cancer Microbiome. And to start off, I'll just go to the abstract slide. So at this point, if you would like to pause the video and read my abstract, you can. But I'll first start off with the background and research questions and the research approach I've used. So to provide a background, the microbiome is a collection of bacteria that reside in the tissues, and specifically in cancer, it's been implicated in a variety of tissues. And the nature of it is such that there are specific microbial effects in carcinogenicity and in inflammation for the different tissues. And in terms of the breast tissue specifically, it was previously thought that it's sterile. However, it's now known that breast tissue has a diverse microbial community where the differences have been observed between the healthy and tumor tissue. And currently, in microbiome research, the taxonomic databases have been so massively expanded and cleaned that they allow for a more accurate and updated taxonomic assignment of the microbes, where specifically pipelines and databases can account for unforeseen biases. So my project will analyze data from 4 studies investigating the microbiota of breast tissue, including papers by Urbaniak, Heekin, Schwann, and Chan. Currently, I'm in the process of analyzing data from the Urbaniak and Heekin papers. And through my research, I aim to address 2 questions. First, how robust are the original findings to the reanalysis with modern tools? And second, are there new biological insights about the disease that emerge from a combined meta-analysis of all available studies? So the research approach I'll use first to kind of address my first question of how robust are our findings. We first need to get the data from the NCBI SRA, and then if the FASTQ files are already merged reads, then we can go straight to the data2 analysis plus phyloseq. Otherwise, if there are forward or reverse reads, then we first need to go remove the primers with cutadapt and then go to the data2 analysis plus phyloseq. And once we get our results from that, we can compare these findings to the original studies. So after this, we can go to the phylogenetic analysis and then principal coordinate analysis. And then when we complete this for each individual study, we can combine our findings from all 4 of the studies into a meta-analysis through multivariate analysis and a random forest application. So this part will help to integrate our findings from each study into a meta-analysis. So my findings from what I've currently done through these preliminary results are that, so looking at figure A, which is an abundance bar plot from the Heekin data that I've analyzed through data2. So the genus includes Aphasia, Bacillus, Coendozoa, Pactobacterium, Serratia, Streptococcus, and Streptophyta. So this is the bacterial composition of the data from data2 from the Heekin paper. And it shows the bacterial composition at the genus level. And this is the abundance bar plot that I've made through the data2 analysis of the Urbaniak data. And at the genus level, it includes Escherichia shigella, Flavobacterium, Lectospiraceae, Mesorezobium, and Salmonella. And then looking at figures C and D. So with figure C, that is the abundance bar plot from the original Heekin study, showing the bacterial composition of the benign and cancer samples at the phylum, family, and genus levels. So they have the taxonomic assignment for the phylum, family, and genus levels. And then figure D is an abundance bar plot from the data 2 analysis of the Hekin data showing bacterial abundance of the benign and cancer samples at the phylum, family, and genus levels. And specifically, I've looked at the genus levels of the Hekin data in figure A, which is enlarged here. So these, in both of figures C and D, the bacterial taxa don't really overlap at any taxonomic level and don't contain any similarities. So our reanalysis of the Hekin data, when we compare our data to the plot from the original study, we don't get any similar results between these two. So they have a very different taxonomic assignment than what our results show. So kind of moving to the recent findings, next steps, and the acknowledgements section. So our current findings show that really our results or findings are very different from the original studies. So the bacterial abundance from our findings from the Hekin and Urbaniak data analysis differ a lot from what is reported in the original respective papers. But it's exciting to discover that our preliminary findings don't align with the original results. So that means that there are biological factors in the updated data sets, so Silva is what I've used, that really no one has explored yet. So both of these data are a few years old. So the Urbaniak paper was published in 2014 and Hekin paper was published in 2016. And the data sets that I'm using are were recently published, so a few months ago. So my updated methods provide newer findings than what was previously reported, which is more accurate and more reliable in that sense. And really the next steps for me are to analyze the Schwann and Chan data and run them through data 2 and phyloseq and implement an approach similar to what I've stated in the research approach section. So going through data 2 analysis and phyloseq and phylogenetic analysis and PCOA analysis, and then moving on to the meta-analysis, which combines the findings from each individual study. And for the acknowledgements, I would like to thank Dr. Burns for his great mentorship and guidance in implementing the code that I've used, and generous funding was provided by the Burns Lab. And then these are just my references, so Urbaniak, Hekin, Schwann, and Chan. So thank you for listening to my presentation.